So this is an interesting publication by these guys, Yu et al., and it's quite recent. The DRRW project has spent significant resources on developing resistance to the UG99 family of pathotypes. And so this guy took all of that body of work, and it was considerable, and published this paper. Not sure if you can see it here. Sorry. We'll get to it. But based on his results, his interpretation of the data in a rather large number of publications, he said the total number of resistance loci, resistance to the UG99 race group, in the A genome there were 37 loci that showed resistance to the UG99 family. Those came from the original Triticum urartu genome. The B genome has 86 loci. Those originate from Aegilops speltoides. And in the D genome, 18 loci that originate from Aegilops tauschii. So you can see here that the B genome contains by far the largest number of loci that give some resistance to the UG99 family. There are, in fact, hotspots of resistance loci across the genome. There are a number of QTL, quantitative trait loci. There are 19 of them on 3BS, 9 of them on 6BS, chromosome 6B, the short arm, 9 of them on chromosome 5BL, 7 of them on chromosome 2BL. And there are qualitative genes. These are genes that would be expressed in the, is that right? These genes would be expressed in the seedling stage. There are qualitative genes, a number of qualitative genes on 3BS, 5BL, and 2BL. So if you look at this region of the genome, 3BS, and this region of the genome, 5BL, and this region of the genome, all of it is on the B genome. So these are the hot spots for UG99 resistance in the genome of common hexaploid wheat. And I just put the title of the article is Yu et al. Theoretical and Applied Genetics, a consensus map for UG99 stem rust resistance in wheat. This paper will also be in your package. So a guy by the name of Mike Pumphrey spent a lot of time with PowerPoint putting together this presentation, and it's a bit dated now. But this is the A genome, 7 chromosomes, the B genome, and the D genome. So in the A genome, these are genes that have been identified to chromosome 1A, and this SR1A, 1R, and there's a QTL somewhere here on 1A. On 2A, there's the SR21 gene is. SRTM gene is here. Here's SRND643, which is a gene that's not named yet, but it shows up in a number of the pedigrees that Ravi has in his program that continue to show resistance to the UG99 family. So this is the A genome, the B genome, and the D genome. Any questions? Comments? So we showed you some examples earlier of cytogenetic manipulation to bring genes from alien species. I think the one that I showed you before was transfer of SR44. This is the gene SR39 being transferred from this, it's called a monosomic addition line, and this is the chromosome that's carrying the SR39 gene, and this is the shortened chromosome that carries the SR39 gene. So we've taken the gene off of this chromosome and put it onto a wheat chromosome. So we can then look at some other species that contribute to chromosomal manipulation. These are the 7 chromosomes of Aegilops tauschii. These, and again, you can't see the credit, but these come from the USDA ARS lab in Kansas. This doesn't look at all like wheat. I'm not even sure what it is. But there's an ability to take genes out of grasses like this that have some agricultural utility and put them into the genome of common wheat. This is a compilation of genes that have been transferred from alien species for resistance to UG99. So this is the gene that has been described, and it is in the gene catalog. This is the genus and species that the gene came from. This is where the gene is located on the chromosome. And the question is it valuable? Does it have any utility in common practice in agriculture? Robert helped me with this, but we were pretty sure this one does, and this one does, and this one does. Are there others that I'm missing? 1, 2, 3 out of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 19. So although we can take genes from alien species and put them into a wheat chromosome, it's not always immediate that there has some agricultural utility. Very often these genes bring with them some linkage drag, we call it. So if we need a chromosome segment that's this big, but the chromosome segment is this big, then there are genes on there. We want this that has only the gene of interest, but there's a lot of other genetic information there that simply is detrimental to agricultural production. Does not contributes the resistance, but it also brings a lot of genes that do not contribute to production. So we've gone through a little bit of the genetic sciences. When I got out of graduate school many years ago, many of these things were not even considered. I don't know. I remember studying population genetics. And the reason I remember it is because this guy wrote a textbook. It was about, maybe Sajid knows this one. It's about this thick. And I think every page has many, many, many mathematical formulas on it. Quantitative genetics or statistical genetics is not the same as population genetics. Molecular genetics, human genetics, evolutionary or molecular genetics again. So we've spent 1 hour here trying to give you a sense of the genetics of the wheat host and how the chromosomes of wheat can tolerate an addition of alien chromosome to it if it's not too large. It has agronomic value. If it's too large, it may not. I'm reminded that the 1B1R translocation included the entire short arm of the Y chromosome, and it became very useful in agriculture. It only became unuseful when that SR31 gene became susceptible to the new virulence that had arisen in Uganda in 1999. Now in the USA, we have a lot of 1B1R still in practice, still in the cultivars of the country. But we don't have UG99 there yet. So that gene still serves a purpose in the US with the current population of stem rust spores, the stem rust population that is there now. And so the real question is not only not just what is the gene that's in the population of the wheat plants in your country, but what are the virulence genes that are in the pathogen population that may or may not render the resistance genes in your varieties susceptible or not. I mentioned a couple of terms about, I called it painting chromosomes with fish and gish. And I'd always wondered a little bit, so this came directly from the internet. Fluorescent in situ hybridization is a cytogenetic technique that allows you to detect and localize the presence or absence of specific DNA sequences on chromosomes. So when you look at those chromosome images with a nice bright green or bright red highlight, that's a function of this technology. An evolution of that technology is genomic in situ hybridization. So when you Google or Bing, when you search the internet for these terms, this is the first one that came up for GISH. It was also a debut album by the alternative rock band, the Smashing Pumpkins. I thought that was interesting. You can find anything on the internet these days. Genomic in situ hybridization is an improvement on this, and I really put this in here for you to understand some of the terms that went into the ability to distinguish different components, different portions of a genome that came from some alien species, perhaps.